Hi class, and welcome to your next video on the second part of cellular respiration. So I know we've talked all about what glycolysis is, the Krebs cycle, electron transport chain, and oxidative phosphorylation, but we're going to go a little more in depth. So remember how glycolysis starts out in the cytoplasm or the cytosol, and we make some ATP. We're also going to be making NADH, which remember is your electron carrier, and your electron carriers are going to be used in the electron transport chain here. Now, because you make two pyruvates, we're going to go twice around the Krebs cycle, and we're going to make one ATP for each turn, but we make a total of two, and we made NADH and FADH2 for the electron transport chain and oxidative phosphorylation. Okay, so glycolysis, a little bit of review. Glucose is converted into two pyruvates. You should already know that. But now we're going to talk about what else we're making and why that is important. So we start out with two NAD pluses, and they get reduced, meaning we are adding electrons to them, and they become NADH. These are going to go to the electron transport chain, and we're going to use that for those high energy electrons which go down the gradient and are eventually going to make ATP for us. So 2 ATP are made by substrate level phosphorylation. So what is happening is we are using an enzyme that adds a phosphate to an ADP, and we're going to make our ATP that way. It does not use oxygen. So bacteria are often going to use this because they do not need oxygen. Also, they do not have a mitochondria, so they cannot go through all of the levels of cellular respiration. So an enzyme transfers the phosphate from some substrate to the ADP to make ATP. So we're going to look at that. So this is a good figure. You should probably copy this down in your notes. You see us starting with glucose, which has 6 carbons, remember, and we're going to make those 2 NAD pluses into 2 NADHs by adding 2 electrons. So you see we get the 2 NADHs too in here. Now we're going to change the ADP into ATP, but how we do that, we're going to have to add a phosphate. So adenosine diphosphate is your ADP. So we're going to add a phosphate to get adenosine triphosphate. When we finish with this, we're going to have 2 pyruvic acids. And remember, the pyruvic acid has 3 carbons each. Now let's look at this substrate level phosphorylation. So you have a substrate that has a phosphate attached. It's going to be the substrate of an enzyme. And the ADP is also going to be part of the substrate of the enzyme. And the enzyme is then going to act upon the substrate, thus changing it into ATP. So we're going to wind up with whatever that phosphate was attached to, an ATP. Remember that ATP is what we're using for our energy. So pyruvate enters the citric acid cycle. Pyruvate must undergo some changes prior to entering the mitochondria. So pyruvate does not go directly to the citric acid cycle, but must be changed to form what we know as acetyl-CoA. The first step is the removal of a carboxyl group, which is COOH, that forms the CO2. So we're going to have CO2 released from this. The second is the removal of 2 electrons from the 2 carbon compounds remaining. So remember, we still have the pyruvate, which we're changing into acetyl coenzyme A. So we're removing 2 of those electrons from the carbon. And finally, the coenzyme A binds to the 2 carbon fragment that we took off before, forming the acetyl-CoA. So what does that actually look like? So if we start over here in the cytosol, you'll see the pyruvate. And the pyruvate, remember, is that COOH. We're going to remove this group right here. Sorry about that. We're going to remove this group right here. And what we're going to do is we're going to go through a transport protein, which we're going to then remove the CO2. Remember, we're going to add on those electrons, so you see the NAD plus becomes NADH. And eventually then the coenzyme A will be introduced. So now we have acetyl-CoA when we get to the mitochondria. So the citric acid cycle, acetyl-CoA, which has 2 carbons, enters. We produce 2 CO2s, 3 NADHs, 1 FADH2, and 1 ATP per turn, which means we produce a total of 4 carbon dioxide, 6 NADHs, 2 FADH2s, which is another electron carrier. Remember, we talked about this. And 2 ATPs. And we're going to do that again via substrate level phosphorylation. Alright, so here is a picture of the citric acid cycle, or it's also known as the Krebs cycle. So we have the pyruvate here, and some of the CO2, some of the carbon comes off as CO2 over here. What we do with that is that acetyl-CoA is now going to enter into the citric acid cycle, and it's going to go through a series of chemical reactions in order for it to harvest all of those electrons and make that ATP. So the 2-carbon acetyl-CoA goes into the citric acid cycle, and firstly, we're going to make that NADHs. We're going to harvest those electrons. We're going to be able to use it. And remember, we're sending that over to the electron transport chain. So then we're going to make our FADH2. Remember, we only make 1 FADH2. And then we're going to have carbon let off in the form of CO2. This is where your carbon goes eventually in the electron transport chain. We are always letting it off as CO2. And then finally, we're going to make an ATP. We're going to go through this cycle twice, because remember, that pyruvate, we make 2 pyruvates, so we can make 2 acetyl-CoAs, and then we can go through the cycle twice. Okay, so oxidative phosphorylation. The electron transport chain and chemiosmosis, they're both part of oxidative phosphorylation. That last step technically has 2 steps within it. The electron transport chain, where, remember, we talked about the hydrogen ion, which went down the staircase, which gave it some potential energy, and then finally it's going to go through a protein at the end, which is going to allow it to make the ATP bond to make the ATP and bond to the oxygen, making the water. So we have NADH and FADH2 dropping off electrons in the electron transport chain. This is where we're going to get those hydrogens. That's where we're going to get the potential energy for our stair steps. And it occurs in the inner membrane of the mitochondria. So we've been talking about chemiosmosis, and now you're going to get to learn all about chemiosmosis. I hope you're excited. So the electron transport chain provides energy to pump hydrogen ions against their gradient. What that means is we're using active transport. Remember when we talked about active transport, you need ATP or energy to do active transport. So what is that meaning? Well, we're not using the ATP that we're generating. We're using the energy from the hydrogen ions going down the protein channels, down that staircase. We're using all that energy it's making in order to pump ions against their gradient. The potential of this concentration gradient is then used to make ATP from a process called chemiosmosis. So whenever we harvest this potential energy and we use it, that's going to be chemiosmosis. We're making that energy via the potential energy that we formed from the hydrogen ions. So the hydrogen ions flow down the gradient through ATP synthase. And ATP synthase is an enzyme in the membrane that produces ATP. Now, how does it do that? Well, as the hydrogen ions are flowing down the gradient, it's actually going to turn. And there's going to be some substrate level phosphorylation going on between ADP and phosphates to make ATP. As it turns, that phosphate is then able to attach to the ATP, making ATP, and then we're going to do that 34 times in order to get all 34 ATP. So if you take a look at this right here, this is your electron transport chain and oxidative phosphorylation. So as we see right here, we have the NADH right here, and we have a hydrogen ion coming off. And that hydrogen ion is then going to be pumped against his gradient, and he's going to eventually come down here. So right here is the ATP synthase. And this is the part that's going to turn, and we're going to have the ADP and the phosphate, and that's going to bond together to make our ATP. Now remember how we said that oxygen was the final electron acceptor? Well, we have this oxygen right here as our final electron acceptor, which is then going to bond the two hydrogens to it. We're going to make water out of that. Okay, I hope you took great notes, copied lots of the pictures, because you're going to need those to study from. Make sure you have all of the detail from these pictures, because we're going to go over them in class, and you're going to need some kind of reference to look at that. Okay, we will see you in class.